Hello, my name is Shinjini Goswami and my ID is 2014-01037. I am going to talk about some of the research related to our group topic, CRISPR and HIV. My area of research in this project was about how HIV-1 has been shown to escape the mechanism of CRISPR-Cas9, and I will use the next few minutes to explain about that. Afterwards, there is also a section on the limitations of CRISPR-Cas9 regarding its treatment of HIV. So now I come to my first topic. Can HIV escape from CRISPR-Cas9? Before proceeding to the main points of this topic, I would like to make a distinction between the two types of genetic sequences, which are conserved sequences and non-conserved sequences. Conserved sequences are series of similar sequences aligned in the same way of molecules such as DNA, RNA, proteins, etc. produced by an organism. The more conserved sequences an organism has, the more it has remained unchanged since its evolution. In the diagram, you can see the conserved sequences highlighted by the arrows. The second type of sequence is the non-conserved sequence, which are essentially mutations induced in sequences over time that help create different genetic patterns, mutations, and different subspecies. In the diagram again, the non-conserved sequences are also indicated by the arrows. HIV-1 is notorious for developing a resistance to almost all sorts of antiretroviral drugs over time. In this short clip, you can see how HIV-1 can replicate and start replicating and begin to complete its entire life cycle just because it's a patient's responsibility to take the proper dosage of the HIV drug, and he has not done so. Basically, that is one of the major problems of HIV treatment. The fact that in spite of there being several drugs to cure HIV in the market, it still seems to manage to develop resistance to any kinds of drugs. Sometimes it's also the patient's responsibility because he may not take the proper dosage on time. However, recent observations have shown that HIV-1, on being cut by the CRISPR-Cas9 genetic scissors, also develops mutations. These mutations are of two types. Some of these mutations are deleterious, that is harmful. This means that these mutations either produce subspecies which are not able to survive, or that the CRISPR-Cas9 guide RNAs can still detect them and cut them off. The second type of mutations are the non-deleterious mutations, that is, which are not harmful. These are the ones which are created in such a way that the guide RNA is not able to detect them. Thus, the HIV virus can create mutations which are non-deleterious mutations and thus escape the scissors of CRISPR-Cas9, as we can see in the diagram. An experiment was performed using HIV-1 virus culture and the CRISPR-Cas9 was used to cut the specific cells, specific genetic sequences. Some interesting observations were also recorded from the experiments. Some of these observations are: firstly, when non-conserved HIV-1 sequences were attacked by CRISPR-Cas9 guide RNAs, the escape was larger in quantity and faster as well. It took longer for HIV-1 to escape from Cas9 single guide RNAs that targeted the more conserved sequences. The second observation was HIV-1 escapes from attacks targeted at interfering into specific sequences by generating mutations. With this kept in mind, it would have been a valid assumption that HIV-1 would change the DNA sequence of the sequence targeted by the guide RNA. In reality also, when the target viral DNA were sequenced, mutations were identified that interfered with guide RNA recognition and helped the virus escape. Thus, the experiment confirmed some valid assumptions. The third observation was an unexpected and interesting observation. The result of the experiment showed that most of the mutations were clustered at the site where Cas9 was explicitly designed to cleave the viral DNA. From the picture, you can see that the guide RNA binding site is a large site with around 19 to 20 gene pairs. However, the mutations were mostly clustered at the site of the Cas9's cleavage site, which is indicated by the red colored oval. The fourth observation is that it has been observed that there were many frequent occurrences of insertions and deletions, which are known as indels. Many of these indels were seen for the non-conserved deep DNA sequences. Deep sequencing and analysis showed that these indels were generated by the cellular non-homologous end joining, that is NHEJ machinery, that repairs broken DNA. Thus, after CRISPR-Cas9 excision, NHEJ generates a number of mutations at the cleavage site, which can be both deleterious and non-deleterious. So, in a nutshell, the cell repair mechanism is also responsible for generating mutations that help HIV-1 escape. Now let me take a short digression here to explain how NHEJ works. NHEJ of double-stranded breaks in DNA is accomplished by a series of proteins that work together to carry out the synapses, preparation, and ligation of the broken DNA ends. 3 steps have been identified in the NHEJ process: DNA end binding and bridging, terminal end processing, and ligation. I will describe these processes in short. First step, DNA end binding and bridging. NHEJ is initiated by the recognition and binding of the Q protein to the broken DNA ends. Q protein forms a ring that encircles duplex DNA, cradling 2 full turns of the DNA molecule. By forming a bridge between the broken DNA ends, Q acts to structurally support and align the DNA ends and protect them from degradation. Step 2. Terminal end processing. NHEJ requires 2 DNA blunt ends in order to join them together. Step 3. Ligation. Once the blunt ends are in place, the XRCC4 ligation complex is recruited to join the DNA ends together. XRCC4 functions as a regulatory element to stabilize DNA and to direct the ligase to the site of DNA breaks through its recognition helix and DNA binding capacity. Now, as we saw, those were the following methods through which HIV-1 managed to escape the mechanism of CRISPR-Cas9. Although no permanent solution has been developed to this, there are still some proposed solutions to decrease the chances of HIV escape. Some of them are: the first kind of solution involves creating variants in the existing CRISPR-Cas9 mechanism. One solution may be to program Cas9 with multiple single guide RNAs that target conserved viral DNA regions, as well as some of the known mutations that HIV-1 has been known to generate. Another solution involves creating new Cas9 variants that are able to cleave DNA outside of the target sequence, so that the mutations created by NHEJ repair will not prevent the guide RNA from binding and excision. It has also been proposed that new CRISPR-like enzymes may also provide such a solution. For example, the newly discovered CPF1 protein acts like Cas9, but unlike Cas9 that cuts the DNA in the region adjacent to the guide RNA binding site, CPF1 cuts in the more distant regions of the target sequence. Another anti-escape solution is the suppression of the DNA repair activity done by NHEJ, which can be achieved with drugs that target NHEJ enzymes. Some of these drugs are the more common anti-cancer drugs which are found in the market. However, this has been found to be potentially dangerous because once the cell repair machinery has been suppressed, it may lead to rapid cell death and susceptibility to attacks. The next section which I want to talk about is the limitation of CRISPR-Cas9 in the excision of HIV-1. CRISPR-Cas9 has been shown to be effective in excision of HIV-1 from acutely as well as latently infected cells. However, none of these studies have tested the ability of CRISPR-Cas9 with regards to excision of HIV from resting CD4 plus T cells of infected people. I would like to mention that resting cells are those cells which are quiet, that is, not currently undergoing reproduction through mitosis. Mitosis is a part of the cycle of cell division. The chromosomes of a cell are copied to make two identical sets of chromosomes, and the cell nucleus divides into two identical nuclei. Before mitosis, the cell creates an identical set of its own genetic information, which is called DNA replication. HIV replication is one major problem in latent as well as infected cells, and that problem has been addressed by CRISPR-Cas9. However, for those cells which are not reproducing but which may also act as a potential host for HIV virus, there is no such research method which has shown what the effect of CRISPR-Cas9 is. This is one interesting area of research that needs to be investigated in future. Thank you for listening.